So in today's video, we have a relationship between glycolysis and pentose phosphate pathway. So basically, the glucose which enters inside the cell, it immediately converts into glucose 6-phosphate. At this point of time, the hexokinase enzyme is working here to take care that the glucose molecule which has entered inside the cell should not escape out, because transporters are available to transport glucose in both the ways. We just want to trap the glucose molecule. So we'll immediately convert, as soon as glucose enters inside the cell, we'll convert glucose into glucose 6-phosphate with the help of the enzyme hexokinase. Now, once glucose 6-phosphate is formed, glucose 6-phosphate molecule and the cell will sense whether ATP is required or not, or the cell should move forward in the glycolytic pathway or not. If at this point of time cell needs NADPH or ribose 5-phosphate, so then there will be an interplay between glycolytic pathway and pentose phosphate pathway. So when the cell needs NADPH as much as it needs ribose 5-phosphate, the cell will carry out the oxidative phase of pentose phosphate pathway. This will generate the NADPH, this phase. Now, when the cell needs ribose 5-phosphate more than it needs NADPH, the cell will transform glucose 6-phosphate into fructose 6-phosphate. And fructose 6-phosphate will then split into glyceraldehyde 3-phosphate via glycolytic pathway. This fructose 6-phosphate and glyceraldehyde 3-phosphate molecules, they will be transformed into ribose 5-phosphate via reverse steps of non-oxidative phase of pentose phosphate. pH more than it needs ribose 5-phosphate, the cell will carry out the oxidative phase, then the non-oxidative phase, and finally gluconeogenesis. So this will help the cell recycle back the ribose 5-phosphate into glucose 6-phosphate, which can be used to synthesize even more NADPH molecule. If the cell needs both NADPH and ATP, then the cell will undergo oxidative and non-oxidative phases, which will help form the needed NADPH and transform ribose 5-phosphate to glycolytic intermediates. Now, these intermediates can then be used to synthesize ATP.